Welcome to this lecture on cell signaling, where tiny molecular conversations have huge effects. Today we'll explore how cells sense, transmit, and respond to information using diagrams that map receptors, cascades, and outcomes. Think of cells as social networks, with antennas and switches. And keep in mind we'll follow signals from arrival all the way to changes in gene expression and medical interventions. Even single-celled organisms can detect incredibly small chemical differences, less than 1%, and they use those cues to move toward food or away from danger. The diagram here shows a subtle gradient with feeding and fleeing zones, and on the right you can see how sensitive molecular antennae on the cell surface convert tiny concentration differences into survival decisions. Signals arrive and travel in different ways, depending on distance and context. Direct contact for juxtacrine messaging, nearby diffusion for paracrine signals, and long-range hormonal broadcasts for system-wide coordination. The slide's illustrations show bound neighboring cells, soluble factors in extracellular fluid, and a bloodstream-style broadcast, emphasizing how the same message can be routed locally or across the whole organism. Neurons combine rapid electrical pulses with precise chemical release to communicate exceptionally fast and accurately. The schematic links microsecond ion fluxes to neurotransmitter release at synapses, showing how an electrical wave triggers a targeted chemical burst. This dual mode explains why the nervous system is high speed, while hormonal networks provide slower, longer-lasting regulation across the body. Receptors are not passive binding pads, but dynamic allosteric machines that change shape to determine signaling outcomes. The graphics show kinase-linked receptors, GPCRs and nuclear receptors, and a central cartoon of ligand-induced conformational shifts. That shape change, plus cooperativity in context, is what gives cells such fine control over whether a signal is amplified, dampened, or precisely tuned. When a ligand binds, the information must cross the membrane, and that's where the cytoplasmic tail twist comes in. The visual shows a receptor flipping its intracellular tail to recruit adapter proteins, initiating a cascade. That twist translates an external cue into internal biochemical steps, launching amplification and branching pathways that carry the message deeper into the cell. Small molecules and proteins broadcast signals with very different speed and reach. CAMP, calcium, and IP3 diffuse rapidly and activate many targets in milliseconds, while proteins travel more slowly for localized effects. The slide also highlights how rapid responses are shut down quickly by enzymes or pumps, so timing and termination are built into the signaling loops for precise control. A faint external stimulus can be converted into a robust internal pulse through a MAP-KKK kinase cascade. The diagram walks through sequential phosphorylation steps that amplify the signal, while feedback breaks keep activation from running wild. This ladder-like architecture turns a small trigger into a decisive nuclear transcriptional pulse, but still preserves tight regulation for cellular precision. Transcription factors act as the final receivers, converting transient cytoplasmic signals into sustained changes in gene expression. Activated proteins bind enhancer regions, recruit chromatin remodelers and RNA polymerase, and open up DNA for mRNA synthesis. The result is that short-lived signaling pulses can be integrated into long-term phenotypic shifts, from altered metabolism to cell fate decisions. When signaling goes awry, it hijacks cellular circuitry and contributes to disease. Mutated receptors can drive unchecked growth in cancer, stuck kinases lead to insulin resistance in diabetes, and misdirected immune signals cause autoimmunity. The pathway maps on the slide illustrate how molecular understanding becomes the blueprint for targeted therapeutics and a structured drug discovery pipeline. Restoring cellular etiquette relies on precise molecular interventions that correct the faulty signals at their source. Examples shown include small molecule inhibitors, like imatinib, that block rogue kinases, monoclonal antibodies such as trastuzumab that prevent receptor activation, and CRISPR-based editors that rewrite defective genes, all aimed at restoring normal communication and cell function. Looking ahead, engineering programmable living microcomputers combines optogenetic actuators, synthetic receptors, and self-reporting, self-correcting cell designs. The slide sketches programmable cells that detect problems, report status, and enact fixes, effectively turning biological circuits into engineerable systems. This new frontier promises precise, customizable control over cellular behavior with powerful therapeutic and synthetic biology applications.